Hey guys, it's Standing for Truth coming to you tonight for another hopefully very informative and profitable broadcast here. We're titling it Refuting Evolution and Defending the Creation Model. Genetics confirms creation. We know this through empirical evidence. Now, I'm going to address some arguments here that have been raised against our model. And I've said it many times before, and anyone listening to my discussions and my videos know that, I definitely hold to a biblical model, a model of pre-existing heterozygosity, pre-existing nuclear DNA differences. In the same way, obviously, that God was not limited to creating static species. We would say God was not limited to creating static genomes, and that all falls in line with the pre-existing heterozygosity. And this means that God may have caused changes by placing intelligently designed genetic algorithms into the genomes of these created kinds. This would have resulted in changes in genetic information and even create what may be seen as new information, but really it's just an amplification or a new trait being emphasized based on this pre-existing genetic algorithm, which means that wouldn't be new, novel, meaningful information as the evolutionists would want us to believe. So obviously, God, according to our model, he did engineer information, significant information, in compressed, hidden form, obviously in our genome, based on decompression through whatever process. This will be seen as new information. Now, obviously, we don't believe that all current human genetic differences are actually due to mutation. The problem that the evolutionist has is they're not making a distinction between mutation and then design variation, because we know for a fact that there are a huge number of single letter differences between people. And we know these are mostly shared among all people groups. What does this indicate? Well, obviously, this indicates that much of the diversity found among people was actually designed, which falls right in line with our model of pre-existing heterozygosity. Now, Adam and Eve would have carried a significant amount of this diversity. And this diversity was well represented on the ark and in the Babel population immediately after the flood. And the post-Babel people groups were definitely large enough to carry away most of the variation present at the Tower of Babel. Now, a common objection that I've seen raised once, the first time was in my second discussion with Jackson Wheat, which I highly recommend listening to. But based on the flood bottleneck and based on the alleles being passed on, they'll say that these population sizes that were dramatically reduced, this wouldn't have allowed for the variation that we say occurred based on that design variation. Now, this is actually not an issue given our model of pre-existing functional DNA differences. And that's why we always need to identify the straw man, because if they don't see these millions, these actually tens of millions of nuclear DNA differences as functional DNA differences, it's always going to be a straw man, because they're never going to be able to understand their model. They're not going to be able to see why their argument is really not an argument at all, because this population bottleneck was very short, which means the heterozygosity of the ark passengers would have no doubt been minimally affected. I think this is quite obvious given our model. Now, for sexually reproducing kinds, a male and a female could have easily possessed a combined, well, it's a fact that they would possess a combined 4 copies of nuclear DNA. These copies could have been very different. And what that means is they would have preserved a massive amount of speciation potential. Now, after the flood, the process of speciation obviously resumed. Migration after this period would have had profound effects on the genetics and the genomes of each kind. Now, these post-flood conditions that have been talked about many times before, given our model, were obviously ripe and perfect for shifts from heterozygosity to homozygosity, which would have most definitely resulted in new species based on small population sizes, the resculpted Earth, migration, genetic drift, and so on. Now, obviously, the alleles is also not an issue, as I've touched on before. If given our model, and if the evolutionist understands it correctly, we're defining an allele as a single genomic position, independent of its relationship to an actual gene. That means enormous allelic diversity can and will be generated by mutation. But as has been said before, allelic diversity need not arise via mutation. Because if we use, and we are using, this genomic position definition of allele, say rather than the gene unit definition, other mechanisms besides mutation will generate allelic diversity. We know that a single gene typically does span thousands of nucleotides, and single nucleotide variants might actually be distributed throughout the gene. For example, at 90 of the nucleotides within the gene, if we allow for the genomic position definition of alleles, which we are, every single one of those 90 single nucleotide variants may have existed in a heterozygous state in each of the individuals of the pairs brought on board the ark. This means that the entire genome reveals a tremendous and unlimited potential for allelic diversity on the ark. Now, obviously, if you've listened to any of the discussions, if you've studied our model is far more superior to the model that evolutionists provide, the model of universal common ancestry. And they scoff at the evidence that we're providing, not even realizing that they believe every single animal we see today ultimately came from 2 animals. Evolution, pond scum to people evolution, really is pseudoscience. And as an aside, before I get into my next point here, I was listening to a discussion the other day on modern day hysteria, I think is the name of the page, between Steve McRae and I believe a Christian apologist named Chris Boyer. And their arguments are so weak and silly. I mean, they're sitting there arguing over definitions of evolution. Now, the word evolution is a very slippery term. We have to define our terms correctly because obviously we see change. Now, a technical definition for evolution is a change in allele frequency or gene frequency or gene pool in populations over generations. Obviously, we know individuals are not evolving, it's populations. But the thing is that change limited? That's what it comes down to. And that don't seem to understand. There's obviously a built-in barrier somewhere. A dog, an elephant, and a mouse are all mammals. But we're not going to get a dog as big as an elephant, and we're not going to get a dog as small as a mouse. Even though there are animals as small as a mouse, like a mouse, there's obviously a built-in barrier in there somewhere. And that is why our model of pre-existing heterozygosity, pre-existing DNA differences, is most definitely the stronger model. Now, in my last discussion, which I highly recommend listening to, titled "Is Junk DNA a Myth?" CRISPR, which I just addressed some of the arguments here, believe that based on the genome sizes of various animals, that pre-existing heterozygosity actually would be an issue because of, I guess there's no better way to put it than genome overload. But what they don't understand, and what he obviously doesn't understand, is that if God compressed this information into our genome, a lot of it is in hidden form. And it's also a revelation of the lack of understanding of the location of biological information. Now, a large storage of this information is DNA, deoxyribonucleic acid, but obviously it's certainly the location of a tremendous amount of it. This gene-centered view is what unfortunately hinders people like CRISPR from understanding the tremendous potential for compressed and hidden information, because obviously the architecture of the cell, say including the cell wall, nucleus, subcellular compartments, and even a myriad of molecular machines, did not originate from DNA, but was created separately and alongside DNA. Neither can exist without the other. This is fact. Therefore, a huge, an unlimited, an immeasurable part of biological information resides in living organisms outside of DNA, which means that taking an organism-centric view actually changes the debate dramatically, because when we actually look to the genome itself, we see it as a hierarchical system, because we see, based on this meta information, this compressed information, these evolutionists really need to get past this naive idea and view that we understand the genome because we know the sequence of, say, a linear string of DNA. Because in fact, all we know is the first dimension out of at least 4 in which the genome actually operates. 1, the one-dimensional linear string of letters. Now say 2, the two-dimensional interactions of one part of the string with another, directly or through RNA and protein proxies. Now, the third would be the three-dimensional spatial structure of the DNA within the nucleus. And 4, changes to the first, second, and third dimensions over time. There is obviously a tremendous amount of information packed into the genome that we have not figured out, including, which they may disagree with, multiple simultaneously overlapping codes. Now, a big discussion took place in regards to junk DNA because CRISPR and other scientists, other evolutionists trained in the field, they underestimate the DNA differences. They assume the rest of our chromosomes don't actually encode traits from regions referred to as junk. That's why they think encoded traits have to be contained in just 1 or 2 chromosomal DNA. But all chromosomes encode for traits, which means there is more than enough room in these original created kinds for pre-existing heterozygosity. And at the same time, the genome sizes could have been quite differently created, because natural selection reduces genome sizes. when they eliminate sequences. So if you're looking at a bat kind today versus a bat kind that was originally created, that genome size could be significantly larger in the past. And now, based on the unscrambling of information and natural selection, these genomes have become smaller. Now, a big argument that he had, just getting a drink here, is that in regards to ENCODE, and CRISPR in my discussion is not the only one, they have a major problem with ENCODE's definition of functional. They actually say that ENCODE's definition of functional is too broad, because we know and we agree that ENCODE define function as specific biochemical activity. Now, this doesn't necessarily mean that they know the function of a given sequence of junk DNA, but that there is some activity associated with it. For example, ENCODE found that the majority of DNA is transcribed into RNA. What is transcription? Well, transcription is just essentially copying the DNA into another form, obviously called RNA. Now, this was thought to be exclusive to parts of the DNA that coded for proteins. Now, the RNA would be read in the cell, and the protein assembled from these instructions. Since junk DNA is non-coding DNA, why is it being transcribed in RNA is the question. Now, many evolutionists think this RNA isn't doing anything, such as CRISPR will say that there's activity, but it's not useful activity. A lot of it is spurious transcription. Many believe it may be one of those evolutionary leftovers, say, that hasn't actually been eliminated from our DNA just yet. But again, it's a worldview issue, because they're arguing and they're in conflict with these ENCODE researchers and scientists, because it has not been shown that this RNA is actually non-functional. These evolutionists desperately fight these results. They're saying and using these arguments to accommodate the evidence within an evolutionary worldview. But from a biblical creationist perspective and from our biblical base model, it would actually be very much inconsistent with a logical, orderly designer to actually design organisms with a lot of non-functional DNA. So this finding that junk DNA is not idle, this junk DNA has activity and may actually be functional, is very consistent with a biblical worldview. And this is a big and major reason why they fight it so significantly, because ENCODE, they found at least 80% of the non-coding DNA was transcribed in RNA, suggesting function. This is what scares them. It has been found that the non-coding regions of genomes correlate with biological complexity better than the protein coding regions. What does this suggest? This suggests that the non-coding regions do indeed carry significant information, probably for regulatory activity. And another important part of ENCODE's research is that they didn't look at the portions of the genome involved exclusively in development, which we know such DNA would only have function then. What does this indicate? The trajectory of discovery favors genome-wide functionality. Now, if anyone's wondering, what some of these functions are, well, non-coding DNA is involved in regulatory functions, when and how fast to make specific proteins, embryonic development, the timings of protein manufacture, staging of proteins, say for construction of molecular machines. Some non-coding DNA is transcribed into short chain microRNA that actually binds to the specific sites in DNA, thereby regulating transcription of those sites. Now, some RNA is involved in the splicing of mRNA before it is translated in the ribosome. It's a fact, and it's obvious. It should be obvious to anybody that we're just beginning to understand the developmental process on a genomic level. How coding and non-coding DNA coordinate when, where, and how structures are sequentially put in place to produce a fully functional organism. All of this compressed and hidden information, these tens of millions of nuclear DNA, functional front-loaded nuclear DNA differences, the pre-existing heterozygosity, perfectly explains the variation we see. And the argument being used is really not an argument at all. Now, if we actually look more at some of these genetic based claims that these evolutionists are using, but actually based on the actual data, the empirical data, fits more in line with, once again, the biblical based model. Now, the evolutionists will claim that human to chimpanzee genetic identity is 98 to 99% similar. And in the discussion I was listening to a couple of days ago with Steve McRae and a Christian apologist, Chris Boyer, and I mostly felt that the evolutionists in that discussion were simply bullying him. But the fact is this: the actual genetic identity is only 88% based on plenty of lines of evidence. They'll also say that humans are genetically closer to apes than to other animals. Now, the thing is, we actually predict, based on our model, nested hierarchies within. Because if we actually look at the young Earth creation model, the fact that God designed the entire universe, including the various kinds of biological life that exist in it, then we would obviously expect to find that life fits a design pattern. We also predict this nested hierarchy. And since humans are made in God's image, because we use the Bible as a starting point, we can get a sense for what kinds of design patterns God might have actually used by simply examining the patterns that result from human designs. Examples of these nested hierarchies abound among the design things in our world. And these evolutionists during these discussions, they hate these analogies. They think they're false, but that just means they don't understand our model, because based on the fact that God designed our entire universe, like we said, we're going to look to these things, these designs that we design as humans, and see and figure out how God may have designed life. Because, for example, designed means of transportation easily fit a relative hierarchical pattern. Sedans resemble SUVs more than they resemble tractor trailers. And all three vehicles have more in common than do sedans and amphibious assault vehicles. The latter two vehicles have more in common with one another than with submarines. And this simple pattern matches the type of hierarchy that we see in biology. Now, another claim that they'll say is, and this is a big one, and I certainly suggest looking at my video titled Chromosome 2 Debunked, where I have this discussion with the scientist CRISPR, but they'll say human chromosome 2 arose from a fusion of two ape-like chromosomes. But the actual data, this purported fusion site, is actually a functional DNA element in a human gene. Now, they'll also say that gene order along chromosomes has no function. Therefore, shared gene order demonstrates common ancestry. But the actual data, once again, tells us that gene order along chromosomes does indeed perform a function. And if you missed my section on junk DNA, go back and take a look. Another claim is that humans and chimpanzees share genetic mistakes, aka pseudogenes. But the fact is, these pseudogenes appear to be functional DNA elements, not mistakes. We speak 1 to 3% of the DNA evidence, and this evidence that they're using is based on such limited understanding of the genome. I mean, 50 years ago, what did we understand about the genome? One protein, one gene, one function. But now we know that these proteins, this phenomenon called protein moonlighting, I mean, these proteins are performing multiple functions. We're understanding that the RNA was very much underappreciated, but now it's doing so many unsuspected things, playing so many unsuspected roles. The more we learn about the DNA, the more we realize that these shared genetic mistakes are actually functional DNA elements and not mistakes. And that's huge for our model because these phylogenetic trees, phylogenetic systematics, relies on this key assumption, it's a false assumption, that these nuclear DNA differences, these shared genetic mistakes, are actually evolutionary leftovers. But if they are functional, front-loaded DNA differences, they can't be used to build these phylogenetic trees. There's even a big one where they say that humans possess the broken remnants of an ancient chicken gene. But we now know through further evidence and through further research that no such remnant exists. Instead, the fragment appears once again to be a functional DNA element. Now, what I want to get into here is, based on our model of pre-existing heterozygosity, we want to get a little into speciation. And in a nutshell, if we have these heterozygous ancestors, an almost limitless variety of combinations of chromosomes, genes, and traits are possible. It's unthinkable. And this same thinking applies to other species. The question is, how do some traits get isolated and become a new species? Well, to make it clear, speciation can occur when a new combination of traits confers an advantage that facilitates having more offspring relative to others, or by migration and isolation of a subpopulation. But can migrations in the wild isolate populations quickly enough to produce tens of thousands of new species in 6,000 years? I'm shocked that the evolutionists still use this argument when, in fact, given our model, given heterozygous ancestors, this is not a problem at all, because we know breeding can do this easily. For example, human breeders have produced 850 horse breeds that have 5 to 6 million nuclear DNA differences. Now, there are between 10 and 28 million differences in nuclear DNA sequences in horses in the wild. So what does this mean? If human breeders, say, can create so many species, starting with relatively little genetic diversity, I think it's pretty obvious to anybody with half a brain that natural selection should be able to generate a few species, starting with relatively greater genetic potential. Heterozygosity, for example. Starting with a large diversity, many different combinations of chromosomes can be generated rapidly. Migration events can isolate subpopulations. The isolated subpopulations would become more homozygous because of inbreeding. Now, DNA variety can be lost due to chance, infertility, small numbers of offspring. Some individuals never mate, and for various other reasons. But the point is, the more homozygous a population becomes, the less it will resemble the original population. Therefore, if you're following me here, speciation can occur when a subpopulation has become isolated and moves towards homozygosity. So if we look at this speciation process, it involves 3 steps. Step 1, formation of genetic distinctiveness. That's important. Step 2, isolation of distinct individuals. And step 3, regrowth of new population. Now, we need to understand that steps 1 and 2 may occur in any order. Now, we actually have data for 300 to 400 mammal species. We're representing 23 mammal orders concerning their gestation times, age of sexual maturity, litter sizes, and lifespans of parents. Now, provided that the founder couples had significant heterozygosity, generating tens of thousands of new vertebrate species in 100 years, say since the flood, from 1,100 pairs of vertebrate ancestors, is actually very plausible. And actually, mathematically, it's very feasible. Now, for evolution, and I'm sure we've all gathered this by now, the ultimate cause of change is mutation. But since the mutation rates are slow, and at the same time, the number of differences in organisms are large, much time would be required to explain the current diversity. I think that's obvious. But for us, and for our model, heterozygosity was built into organisms from the beginning, say by God. That means long periods are not required for the production of new species. If you're all following me here, including the evolutionists, and I think anybody with an open mind can see how much more superior our model is, because starting with heterozygous organisms, visible distinctiveness in offspring would soon be apparent. Because in our model, recombination and gene conversion will undoubtedly provide many new varieties of combinations of traits quickly, since as I've stated before, these differences are already built in. Now, to cap this off, we have these, based on our model, we have these testable predictions coming true. And we see this actually with Darwin's finches, because they're further confirming the predictions made by this model. Because when we actually look at a study of Darwin's finches, we observe that a species formed from a breakaway population that was actually founded by a small number of individuals. And I'm going to post the article here to actually provide the citation. But despite being in the same physical location or on the same island as one of the parental species, the new species maintained its breakaway status through reproductive isolation. Since this species was founded by a small number of individuals, inbreeding naturally occurs. But when you actually look at the homozygosity levels, the paper says that a gradual increase in homozygosity was then observed over the next 5 generations, as expected from the small number of breeding pairs, causing genetic drift. Now, in other words, the parental species was relatively heterozygous, as our model states, and the new species shifted to a state of increased homozygosity, just as our biblical model predicts. Now, I'm sure there's always going to be new challenges coming forth, but the challenges that I've thus seen so far are certainly weak. They're certainly not sufficient. And I think it's pretty clear that our biblical model, for one, is a competing hypothesis, but it also makes testable and falsifiable predictions. Pond scum to people, evolution is dead in the water. There's absolutely no evidence for this type of evolution. There are limits. We see decay. We see degeneration on a genomic level. And the change we see is obviously limited. There's a barrier. And our explanation for speciation based on these heterozygous ancestors is more than sufficient to explain the biodiversity we see and know. Okay, guys, have a good night. I hope you enjoyed. And this is Standing for Truth.